Hello everyone, my name is Adam Farmer and I'm a first year Maths PhD student. My project is entitled Mathematically Understanding the Movement of a Cell Given Various Substrates and Stimuli. Aims and objectives. Our aim is to use mathematical modelling to replicate cell motion, specifically how a cell reacts to external stimuli. We use a system of equations which describe how points on the cell's membrane and nucleus behave and interact with each other in external forces. Using a variety of numerical methods, we aim to find the most accurate and reliable way of predicting a cell's motion. The end goal is to compare this to a real experiment and show the same result. Studying experimentally and predicting the migration of cells can help predict the spread of cancer cells in a patient among other applications. So in the following slide we have two pictures. The one on the left is cell migration under a microscope, and the one on the right is the model we are using. So the model we're using in setup is a system of nodal points. These define the outside of the cell's nucleus and membrane. So essentially the nucleus, outside the nucleus, as well as the outside of the nucleus to the outside of the membrane are connected by a system of springs. These allow us to model elasticity. Every nodal point on the membrane and nucleus as well can react to a signal gradient, which I will cover in a minute. How we went about it. Once we had the base equations and considered all forces involved, we had to consider the biological properties of our cell. As mathematicians, we had a number of variables which needed to be accounted for, each of which represents a coefficient related to how the cell reacts in the real world. This means that we can change the variables depending on the type of cell we are trying to model, whether it be a liver cell or a blood cell. This being said, we can produce a time-step model of a cell's motion. On the following slide, you can see two videos. The one on the left is cell migration under a microscope, and the one on the right is a model we managed to produce. So what you're seeing here is a signal gradient coming in from the right, that's diffusing outwards, and the cell is being drawn towards it. So each of the points, as stated before, on the cell's boundary and membrane are reacting to the signal gradient depending on how close they are to it. Conclusions of future work. Using the governed equations and a multi-step method, we can produce a model of a cell's motion and use its current position to calculate the next. This means that we can start to accurately show where a cell will end up given a specific set of initial conditions and a set time interval has passed. In the future, we plan to update the model further to include multiple cells which can collide and the challenge of making it three-dimensional. Thank you for listening.